before I start, this is linked to the slides. You can go and download them now. They're there. Okay. Hello, everyone. My name is Andrey Lando. I'm from Moscow, Russia. I'm going to speak about my team's solution on the ENCODE Dream challenge of transcription factor binding. Here we are. We're seven. In the middle, there is Cheburashka, our team mascot from children's books and Russian cartoons. And I'll later say why. So yeah, this is the challenge. 200 base pair intervals, transcription factor, bound, unbound. I'm going to refer to these intervals as just genomic intervals or intervals in my talk. So this is how the perfect bound region looks in our imagination. So it has highest DNA accessibility. It has motif heat somewhere inside. That's what we're looking for. So this is our data. And what's important here that for most transcription factors, we had multiple training data sets. Yeah, this is data provided by the organizers we're allowed to use. Experimental data, RNA-seq, DNA accessibility. We didn't use DNA shape in our solution. We believe it's scored by our motifs. And yeah, that's it. We used machine learning approach, so we describe each interval with a number of features. It's a binary classification problem. And later, in the end, we use XGBoost, extreme gradient boosting framework for doing machine learning. I'll talk now a bit about the features we used. So first comes the motifs. Given a ChIP-seq peak, one can extract genomic data under this ChIP-seq peak and build a mathematical model out of it. We use ChIPMonk, our motif discovery tool. It yields simple position weight matrices motifs. But yeah, that's rather good. And of course, transcription factor is not alone in binding DNA. It has cofactors. And we used RECOMB database to get cofactor motifs out of it. So in the end, we count motif hits in a region. We have the score of the best motif, and we have best score of cofactors. That's our motif features. So from DNA accessibility data, we have mean, we have interquantile range, and we have some score that describes the fuzziness of the profile in an interval. We also had some features that tried to catch footprints in DNA accessibility data from bound transcription factor, but I won't stop on that. Transcription factor is known to bound promoter regions and to affect translations. So we have a distance to nearest gene, we have expression of nearest gene, and we have dispersion of expression of nearest gene across given cell types. Yeah, that's our features from expression and annotation. The problem we're solving is a problem of highly unbalanced classes. Bound regions are much more rare than unbound ones. But not only that, number of bound regions for one transcription factor across different cell types is significantly different. And we don't know what is the number in the target cell type. These scatter plots show for one transcription factor and two different cell types as like a distribution of bound and unbound regions, bound are green and bound are blue, for two main features, for two of the most descriptive features we have. This is this is best motif score and DNA accessibility. And what we can see from those scatter plots is that transcription factor preferences, they are different in different cell types. And that's a challenge, because given the left picture, try to predict the right one. That's hard. So that breaks machine learning paradigm that training and test data set should be statistically the same. We have to deal with it. So the way we deal with it, the first, we perform unsupervised feature normalization, and I won't stop on that. And the second, for each transcription factor and for each, for each transcription factor and for each target cell type, we can select the proper cell type for training, the most similar one. So the first question is, can we use our biological experimental data to get the most similar transcription, the most similar cell type? It turns out that no, we can't, because different experiments yield different similarity of cell types, and we don't know what to rely on. Matter of fact, it turns out that cell type similarity is transcription factor specific. So we can't use just a biological experiment of the cell type to estimate similarities to get the best cell type for training. Now I'm going to talk about how do we select a proper cell type for training. So given any classifier on the binary classification problem, one can compute a positive likelihood ratio of this classifier as a portion of positives extracted by a classifier divided a portion of negatives extracted by a classifier. And given a classifier that uses just one feature to perform classification, that just sets a threshold and filtrates some elements, one can compute a positive likelihood ratio for this feature, and that will describe a descriptive power of this feature. And that's what we do. So for some features we use, here is positive likelihood, this is positive likelihood ratio. So say for a motif, setting a threshold 50 and filtrating all the intervals that passes this threshold, and computing the ratio of percentage of bound and percentage of unbound there, and setting a logarithm will result in something like 3. What's notable here is that positive likelihood ratio rises almost monotonically as we increase the threshold. These curves are, we call them enrichment curves. And another important observation we made is that for one transcription factor and for one feature, the enrichment curve shape is almost the same. And it's only a matter of a linear transformation. So the idea of selecting the proper cell type for training is to estimate the enrichment curve of the target cell type for each feature we have, and among training cell types we have, select the one that has the closest enrichment curves. So how do we estimate the enrichment curve for a target cell type? As I said, it's only a matter of linear transformation. So to estimate the enrichment curve, we are required only to estimate the enrichment at one point, any point for any threshold. The point we choose to estimate the enrichment is here. So it's closer to the right of the threshold. So it has a rather high threshold. It has a high threshold is one property required for that. And the second property is it should contain enough intervals to make our estimation robust. If we would shift this threshold even higher, we would have 2 less intervals to make our predictions robust. So how do we estimate the enrichment in this point? It turns out that to do that, one is required to build a classifier that yields rather robust predictions for a small number of intervals. So we are required to build a classifier that yields a rather small number, but the rather small number of bound regions, but the percentage of true positives there, of true bounds there, is rather high. And it turns out that it's very easy to build such a classifier, and our classifier is very naive, and so for naivety, it's called Cheburashka. How does the classifier works? It simply sums the enrichments of the features. So by learning, it learns the enrichment curves. And when predicting for each interval, it yields sum of the enrichments of the features it has. So this classifier believes in independence of each features. The algorithm of estimating the enrichment curve is the following. So say we want to estimate the enrichment for a motive. We extract the motive from the training, so our classifier doesn't learn on the motive features. We train the classifier on all the other features we have. And then, given predictions of the classifier, for intervals that pass, for intervals that passes the threshold, we compute the enrichment, and so we know the enrichment at the red point. That's it. When we want to estimate, say, DNA's enrichment, we add the motive back, extract on DNA's features from the training set, and repeat this procedure again. It turns out that we can't really make a really exact prediction of the enrichment. But what we can do is we can order the importance of features we have. And it's important to select the training cell type that has the same order of importance of features. We can't do that in 100% of the times. When we're not sure, we select two training cell types, we train models individually, and then we average predictions among them. So that's our workflow. We do normalization, we do feature engineering, we have naive classifier predictions, we select the training cell type, we use XGBoost, powerful classifier, to make our several predictions, and sometimes we average our training cell types. That's it. I want to discuss our results a bit. So here is the picture for Jaunt, but it's almost the same for every transcription factor we have. So on x-axis, this is our predictions. So all points share the x-axis. On the top graph, we have DNA accessibility, motive hit on the bottom. And what turns out that we can't catch some of the intervals that are really bound. So they have low DNA accessibility, they have poor motive hit, and we just don't have feature that describes them. We don't know why transcription factor bounds there. To significantly improve our performance, we have to somehow have a feature that distinguishes those intervals from the other ones. That's it. I'll be here at the poster D19. These are slides again. In the end, I'd like to thank my team. It was great working with you guys. I'd like to thank the organizers. Thank you. We enjoyed participation in the challenge a lot. And I'd like to thank you for your attention. Thank you.
A nice talk. Two questions. First one, a small one. Did you register that domain name specific for this competition?
No, it was before.
Okay. So my question is about the cell type specific feature engineering. So you mentioned that there are several features that you haven't incorporated into your predictors, right? Like including like shape information, right?
Yeah.
Do you expect when you incorporate those information, you can reduce the dependencies on the cell type specific engineering part?
So, like, if I got your question right, to make the cell type specific features transferable from one cell type to another, we do feature normalization.
Okay.
I don't have an example to that. I think, like, if you come to the poster, I'll explain you how we do that. The point is to make statistically these features the same in the train and the test data set as good as we can.
Okay. Thanks.